In this R&D spotlight, we're speaking with Dr. Ed Boas, one of the lead researchers at City of Hope, who has been pushing the boundaries of what is possible in drug delivery with microscale 3D printing. So before I jump into any questions at all, why don't you tell us a little bit about you, your background, and then, of course, this liver cancer study that we'll be talking about today.
Yeah, so I'm an interventional radiologist, and what that means is we do minimally invasive cancer treatment procedures. So, for example, we can get a catheter into the artery that goes to a tumor, and then we can deliver drugs, radiation, devices through the catheter into the tumor. And we also have special needles that we can place into a tumor, and we can inject drugs. So the trend is really towards providing less invasive and more targeted therapies that have, are more efficacious because we can target exactly the tumor we want to target, and fewer side effects because we're not injecting stuff that goes everywhere. We're sort of targeting the tumor.
So with this liver study that I was reading about, which was the impetus for this interview, can you talk a bit about that particular study?
Yeah, exactly. So we had this idea that, so for example, when patients get chemotherapy, they often get a port to get chemotherapy. So a port is a device that goes under the skin, that goes into the vein, and it sort of provides a more convenient way to get chemotherapy. So instead of getting poked each time, trying to find your vein, you can access this port, which is basically an artificial vein, and it makes it easier to get chemotherapy over multiple sessions. And the idea that we had was when you inject chemotherapy into the port, the chemo goes everywhere. So you're trying to just get it into the tumor, but you're actually delivering it everywhere, which results in side effects because the drug is going sort of to off-target sites. So we had this idea that we wanted to make sort of a chemotherapy port that delivered the drug directly into the tumor. So we ran into a few problems. One problem was, we're frequently trying to target tumors in the liver. And if you put a catheter in the liver, it doesn't stay in the liver because the liver moves up and down when you breathe. And so the catheter actually gets pulled out with breathing motion. So that was one problem. The other problem is if you just put a catheter into a tumor, the drug only goes out of the tip of the catheter. And so if you have a tumor that's larger and your catheter is just delivering to one spot, you're not covering the whole tumor. So that was sort of the problem we were trying to solve, is to design this catheter that you could put in a tumor, it would stay in the tumor, and it would sort of uniformly deliver drug throughout the tumor. And the catheters that were on the market didn't really work for this. And so we had to sort of come up with a new design. And we were going, we were drawing all of these different designs, and we could solve it on paper, but we couldn't make the thing because the design was too complicated for traditional manufacturing techniques. So then we started looking into 3D printing. We had these CAD designs, we could just 3D print it. And so we tried sort of conventional 3D printing, and the details that we needed were too small because, we're trying to be minimally invasive. So we want to make the catheter as small as possible. We want to make the incision as small as possible. So we wanted to scale down, and we had all of these fine details that we needed for this catheter. So that's where we found the microstereolithography was actually able to manufacture the catheter, exactly as we envisioned it.
So do you know anything about any of that material selection in, I mean, I imagine you have to be very careful about the materials you pick to put in a body. So can you talk about that?
Exactly. So it does have to be biocompatible, and both in terms of toxicity, obviously it has to be non-toxic, but also in terms of the body's response to the material. When you put foreign materials, when implanted medical devices, there's often a layer of scar tissue that develops around the device, which can reduce the effectiveness of the device. So yeah, so obviously you have to use something that's biocompatible. For 3D printing, it is somewhat more limited. We're basically limited to what the biocompatible options are for the 3D printer.
So how did the prototyping go for this device?
Yeah, so the prototyping is we start in the lab, and then we go to large animals, and then we go to humans. So, sort of rapid prototyping and iteration is really important because we don't actually know what's going to work. And we had some surprises along the way in terms of how do you make this catheter so that it delivers the drug uniformly through the tumor. So we tried a few different designs. We initially just tested, you stick this catheter in a block of gel and see how uniformly does it deliver the drug. And then once we have a design that works well in the lab, then we can take it to large animals, which is what we published in our paper. And based on that, we're now trying to move it into humans.
In our next segment, Dr. Ed Boas talks about the specific design techniques and best practices that he might recommend for engineers aiming to leverage 3D printing. And he also will look ahead about the most exciting opportunities for 3D printing to enable new delivery formats on multifunctional medical devices.